We are going to describe ubiquination in 5 minutes. Here are the basic components of a protein. Several amino acids, shown here in blue, form a chain. This results in a carboxylic terminus and an amide terminus. These amino acids can interact with one another to form complex structures. This is mostly achieved through hydrogen bonding, shown here in red. If all the amino acids associate correctly, then a properly functioning protein is folded. This protein can be part of a group that accomplishes a function. However, if a protein does not fold correctly, it can prevent other proteins from functioning properly. Sometimes a lot of misfolded proteins will bunch together and form an aggregate. This can lead to detrimental effects such as Alzheimer's or Parkinson's disease. What does a cell do with misfolded proteins? It can send them through a chaperonin, which provides a good environment for the protein to fold properly. Sometimes this does not work. Another route the cell can take is to use a proteasome and recycle the amino acids. This requires a unique signaling molecule called ubiquitin. Ubiquitin is a 76 amino acid polypeptide chain with two notable amino acids, lysine 48 and glycine 76. It is highly conserved in all eukaryotes and is so important that it led to the 2004 Nobel Prize in Chemistry. This was awarded to Aaron Chekhanover, Avram Hershko, and Erwin Rose for the discovery of ubiquitin-mediated protein degradation. We know that ubiquitin attaches itself to a misfolded protein, but it does not do this directly. Instead, it follows a highly regulated process known as the ubiquination pathway. Once the ubiquitin is bound to the protein, more ubiquitin can attach and form a polyubiquitin chain. First, ubiquitin interacts with the ubiquitin activating enzyme E1 in an ATP-dependent reaction. This forms a thioester intermediate at the glycine 76 terminus. This process involves the formation of an adenyl intermediate. Ubiquitin attacks the closest phosphate with a carbonyl oxygen. The E1 complex can then attack the carbonyl carbon on glycine 76 and form the thioester intermediate. The last two complexes in the pathway are the ubiquitin conjugating enzyme E2 and the ubiquitin ligating enzyme E3. E2 contains a sulfur that will bind to the glycine residue on ubiquitin. E3 contains a protein binding site for the misfolded protein. E2 has a higher binding affinity than E1 and displaces it, forming a thioester bond. The ubiquitin is now primed and ready to act on a misfolded protein. Oh look, here's one now. It binds to the protein binding site and exposes a lysine residue, which can attack the carbonyl carbon and displaces the E2 complex. Furthermore, the ubiquitin exposes yet another lysine residue that can interact with more ubiquitin. As each additional ubiquitin binds, it exposes a lysine residue, which promotes binding for more ubiquitins. Binding for at least 4 ubiquitin results in protein degradation. The protein will unbind from E3 and make its way to a proteasome to have its amino acids recycled. These amino acids can then be reassembled into a new functioning protein.